I always wanted to become a doctor, and I was signed up to become a doctor in medical school in Munich. And then last minute, literally last minute, I changed to an engineering class. Never regretted it. But yeah, so I was always very close to that field. And together with my co-founders, Maciej, who is a medical doctor and a genomics researcher, and Maxim, who is an AI researcher, we looked into this field of the intersection of AI and pharma and got really excited by those new types of models coming out, those bio foundation models. And so decided to kick off Helical from there. A foundation model, to make it very simple, is basically a very large pre-trained AI model. Pre-trained meaning that it is being trained once, and then you can use it for multiple things, that has learned on very large amounts of unlabeled data. Unlabeled is key here, and I explain what unlabeled means, but it uses a technique called self-supervised learning to achieve this paradigm shift. And it differs quite a lot from the more traditional AI algorithms, where the AI algorithm actually needed that labeled data to predict something very specific. Some use cases we are currently addressing with our customers, they really go across the entire drug discovery stage, from things such as target identification, so using those models in very low data environment to find better targets, biomarker discovery so that you can stratify your patients for the next clinical trial, knowing the gene signatures that are responsible for success or failure. There's not one model solves it all for many reasons. You need multiple modalities, right? You don't solve problems on one modality, but you could still argue that you can train across modalities over time, right? We actually have a benchmarking framework where for each use case that we do, we look at all the existing models that are out there before we choose one. And funnily enough, it is often a different one coming on top that is very good at a bit more specifically your use case that you're targeting. I think the end goal here is to go towards a virtual cell where you leverage all of those different foundation models across those different modalities. And you start combining the embedding spaces. This is basically the output of the foundation model into one digital cell where you can do all of the things you just mentioned. You can leverage it to do those perturbations that you have in mind. You can simulate it to be a diseased cell, and you can see in silico how you could heal that cell, basically. And this is all possible with foundation models. I think we often work with computational biologists that know a lot of traditional methods. So there is still, and more than fair enough, a certain skepticism against, hey, can those methods actually outperform the state of the art? So how we react to this is really, in all the benchmarks we do, we always include the state of the art methods, that more traditional methods, into the evaluations as well, because we need to prove still that AI models can reach those levels. We make it very easy for a customer to get into a lab in the loop type of setup by bringing back feedback we generate directly in the model and really making it their own model. We really focus on being the platform for the model. Again, meaning making it super easy for pharma to leverage those models in their very specific use cases. What we are not trying to do is leverage the models, for example, to advance our own pipeline, right? So we are not, in that sense, a tech bio company, but we equip the tech bio companies of tomorrow to be the most efficient at building AI and to get the best results. I would like to mention here is that we take an open source approach. This means that we bring parts of our platform on GitHub so everybody can use it and benefit from it. So I think it's something that anybody who is interested in exploring biofoundation models should check it out, because it's really made very beginner friendly. You can simply run very simple example notebooks, and we see many academics using it actually. It's all about speed of iteration. And the more you can do in silico, and I think that's what those models will help us doing, the faster we can be and the more impact we can have on bringing drugs to market faster. And I think it will happen. It is happening. So that's quite exciting.